showed you what you can do with the variant effect predictor, or VEP. I know that yesterday we didn't have much time, so I hope you did get a chance to go through the exercises for the ensemble VEP. So before we begin with the BioMart session, I just want to ask if you have any questions, or if there's anything you want me to show you or demonstrate, or just any general comments. I'll also check the living document. Actually, I will share all of the URLs with you. So we've got the training page, and we've got the living document. And that's in the chat for you now. So I'm going to check the living document if there are any outstanding questions. I think I've answered all of them. Here we go. Okay, so we have one outstanding question, which I will also write down, but I'll also explain it now. So the question in the living document is, why does Arabidopsis thaliana not have any cultivars? So the only plant species we have cultivars available for are rice, barley, and wheat. We don't have any other cultivars for Arabidopsis. I'm not sure why. So in that case, you can actually email us directly, and you can ask us to add any specific cultivars that you're interested in into ensemble plants. So often the way we decide on what species we include in ensemble is the interest of our users. So I'm guessing so far no one has mentioned that they wanted different cultivars for Arabidopsis, and that's why we don't have them currently. But if you do have an interest in them, you can email us, and I'll post the email address into the chat. So you can email us. I will talk, or me or my colleague Alina will talk to the relevant teams, and we can look at how or when, or if it's even possible to add any other species, any other cultivars for Arabidopsis. Then we've got another question from Ajay in the Zoom chat. So Ajay is asking what the difference is between cDNA and protein. So the cDNA is the DNA sequence. So this is the A, T, C's, and G's, G bases, whereas the protein is made up of the amino acid sequence. So the one starting in methionine and then so on. So the cDNA is not more useful. It's especially useful if you are looking at comparative genomics data. So if you're comparing genomes between different species. Protein data is very useful if you want to look at the protein function, what it does between the different species, and so on. The next question is, can we do COG analysis in ensemble if we have UniProt IDs? I don't think we have any KEG information in ensemble. I will need to double check that. So if you could you add that to the living document? I will need to do some digging on that. And then I can give you an answer on there. Thank you very much. And then the final question we've got is, can we perform Ka and Ks calculation in ensemble? I'm not sure, actually. Again, can you put that in the living document? I need to double check that. I don't think so, but I might have missed something. Okay. So let me check the living document again, just to see if there are any other questions. All right. So no other related questions. In that case, I'm going to hand over to Ian. So I'm going to stop my screen share. And Ian can take over from here.
All right. Hello, everyone. Hi, everyone. My name is Ian, and today I will be taking you guys through the final part of the workshop, which is on Biomart. So today we will cover firstly what Biomart is. I will talk about how we use Biomart. I'll briefly introduce the R package for Biomart, which I'll touch on later. I'll show you a couple of links where you can read more about Biomart. I will walk you through a live demonstration of how Biomart works on Ensemble. And finally, there are some exercises for you guys to go over to practice what I've talked about today. So we will first talk about what Biomart is. So to put it very simply, Biomart is a tool that is built into the Ensemble browser. What it does is it allows you to export and query data on Ensemble at a much larger and faster rate than what you can do on the website. You can build quite complex queries with only a few clicks of a button, and it doesn't require any programming knowledge in order for you to use Biomart. And Biomart lets you generate a ton of data in different output formats and different files as well. So you may have a question, which is, why do we want to use Biomart? Why would I not just use the website? Well, we can think of the Ensemble website as, or accessing different bits of the Ensemble website, like different grocery shops. So you may you may type in a specific gene to retrieve information about that gene. You may then go to the transcript section to fetch information about that transcript. And that's a bit like going to the bakery to get some bread, and then going to the cheese shop to get cheese, and then going to the grocery store to get vegetables. Whereas Biomart is kind of a superstore, a one-stop shop. So you can just go to Biomart and retrieve all the information you want for everything you need in one go. So it's much faster than just clicking through the website, and it lets you export a lot more data in one go than you can do on the website. So Biomart lets you do a lot of different things. And here are just some examples of what you can do on Biomart. So you can do things like gene ID conversions between different gene assemblies for a species. You can query different genes at different genomic locations. You can download cDNA, DNA, protein sequences for a bunch of different gene IDs that you have. And as I've mentioned, Biomart lets you export a very large amount of data. And if you were to do the same thing, if you were to query the same amount of data on the website, it would take you much, much longer. So Biomart, as I've mentioned, is built into the Ensemble browser. So this link here will take you to Biomart. Alternatively, you can just go into Google, type in Ensemble Plants, click on the Ensemble Plants link, and there is a button at the top called Biomart. Click on that button and it will take you to Biomart itself. So Biomart is available for most Ensemble platforms. It's available for core Ensemble, so that's for things like human, mouse, pigs, chicken. It's available for Ensemble plants, as well as the metazoa, the protists, and the fungi. But Biomart is not available for Ensemble bacteria. Here, I will talk about how we actually use Biomart, and it's very, very simple. We can boil the process of using Biomart down into 4 major steps. And I will talk about each step in a little bit more detail in a bit. But here's just an overview of how it works. So first, we select our data set, we choose a database, and we choose what species we want to look at. We then apply a bunch of filters to our data set. In step 3, we decide what data we want to retrieve, as in what data we're actually interested in. And step 4 are the results, where we choose how we want our results to be sent to us or how we want the results to be displayed. So I'm going to talk through each step in a little bit more depth now. Step one is the data set. So on Biomart, there are two main databases you can look at. You can either look at the genes database or the variation database. So the genes database contains information for all things genes, transcript, protein related. And the variation database contains all the information regarding any variation associated with Ensembl. And then in step one, you also choose what species you're looking at. And there will be a drop-down button where you can select all the different species that are available on Ensembl to query. So that summarizes step one. Step two is then we apply some filters to our data set. So step one gives you a ton of information. It gives you all the information that exists for that species you're looking at. But that's too much information, and we want to apply some filters to filter out bits of information that we want. So we can filter our data set for things like region. So for example, if I was looking at Arabidopsis, I may only be interested in one of the chromosomes in Arabidopsis. So we can choose chromosomal region or chromosome number as a particular filter. If I have a list of genes, or if I have a specific gene in Arabidopsis that I was interested in, I can apply that gene as another filter. I could also apply a functional Go term as a filter. So for those that aren't aware of what Go terms are, all genes have functions, and a Go term is essentially a word that is associated with each gene that basically describes what the gene does. So for example, I could be interested in genes that are responsible for cell repair, for example. So I could filter for that Go term in order to retain only genes and information associated with that function. So that summarizes step two. It's basically a process where we apply filters to our data set. Now, as I've mentioned here, you can apply, I've only listed 3 examples of filters here, but in reality, you can apply a lot more filters than just 3. And what happens is Biomart will only return data that satisfies all your filtering criteria. So for example, we can think of filter 1 as maybe location. So chromosome, let's say filter 1, we want to filter by chromosome location. Let's say chromosome 1. And filter 2 would be, let's say you have a list of 50 genes you want to filter by. What gets returned will be all the information associated with genes that are within your gene list and also within chromosome, are located on chromosome 1. So the more filters you have, the less data will get returned because it's becoming more and more specific. Step 3 is we choose what attributes you want. So this is probably the most important step. This is where you decide what data you actually want. What data do you want to receive from Biomart? And again, just like filters, there are many different options you can choose from here. So you can retrieve things like gene IDs. You could retrieve genomic or genetic features associated with the genes. You could retrieve any variants associated with the genes. You could also retrieve things like orthologs or paralogs. These are sequences that are closely associated with a particular gene of choice. And you can also obviously retrieve sequences themselves, be it whether that's DNA sequences or protein sequences. And finally, we come to the results section. So Biomart will generate a preview of your results table, and you can have the results, you can save the results in a bunch of different ways. So firstly, you can take the option to choose to save it as an HTML, and Ensembl will email your query, your results to you if you put in your email address. You can also save your results in a tab separated file, so that's TSV, or a comma separated file, that's CSV. Or you could also save your results as an Excel file, so that's what XLS means. If you want to retrieve sequence data is only available to be saved in a FASTA format. So that's a very, very standard text-based format for storing genetic sequences. So I've talked about the 4 major stages of how to use Biomart. If you want to read more information about Biomart, there are 2 links here that take you to the papers that describe the publication of Biomart and how Biomart works in a bit more detail. So I'm going to walk you through a live demonstration now of how Biomart works. And for this, we will be looking at genes on chromosome 5D between these 2 genetic coordinates in Triticum aestivum, which is wheat. And we are looking to retrieve NCBI gene IDs, functions, so Go terms, and we're also interested in retrieving cDNA sequences. So I will navigate to Biomart now to show you guys how we can do that.
No, still not.
Oh,
I
it's
still
not.
see your slides.
All right. There we go. So if we navigate to Ensemble Plants, and if you remember what I said, Biomart is available in the top bar here. So if we click on Biomart, oh, whoops. Cool. So this is Biomart. So remember step 1, we have to choose our data set. So if we click on this Choose Database tab, we can see that you can either choose the genes database or the variations database. So we are interested in looking at genes in wheat. So if we first select the genes database, we then have to choose our data set. So these are all the plant species available on Ensemble Plants. And we are interested in Triticum aestivum, which is bread wheat. So if we scroll down, we can see there are many different Triticum aestivum options available here. All of these here are wheat cultivars. So things like Cadenza, Claire, Jaeger, Yuli, these are all cultivars of wheat. We're interested in the main, the primary wheat assembly, which is this one here. So Triticum aestivum genes. What's in brackets here, IWGSC, is representative of the assembly or the organization that made the actual wheat assembly. So if we select Triticum aestivum genes, we can see that on the left side here, we have a data set. So we can see that it's confirmed that we are selecting genes from Triticum aestivum. We now want to apply some filters. So if we click on the filters tab here, if we click on this button here, so before that, these are all the things that you can filter on. You can filter by region, gene, ontology, multiple species, domains, and variation. So for our demonstration, we want to filter genes between 2 different genetic locations. So we click on this button here to expand the region button. So on the slides, we are looking at genes on chromosome 5D. So first things first, we want to choose this chromosome scaffold button, and then scroll down and click chromosome 5D here. And you can see on the left, this filter has now been added to our filter section. So we know that this is going to return only genes associated with chromosome 5D. The second parameter we want to filter by are the genetic coordinates. So tick the coordinates button here. And on the slide, we want to filter between 19.4 million and 21.3 million. Make sure that box is ticked. And again, as you can see here, these start and stop coordinates have been added as a filter. So here there is a count button. And if we click count, we can see that out of 120,000 genes on the wheat genome, when you apply these filters, so on chromosome 5D between these 2 genetic locations, there are only 6 genes that satisfy this criteria. So it's a good idea to use the count button quite often when you're building a query, just to make sure that the number that gets returned makes sense. For example, if it's set 0 out of 120,000 genes, then you know something's gone wrong in your filtering or something hasn't been applied properly. Cool. So in terms of the filtering process, that's it. We only want to filter for genes on chromosome 5D and between the 2 locations. We now need to choose what data we want to actually retrieve from Biomart. So we then click on the attribute section. And if you look at the slides, we want to retrieve NCBI gene IDs, functions, Go terms, and cDNA sequences. What's important to note for the attribute section is here, if I want to retrieve things like sequences, you need to click the sequences button here. And if you want to retrieve things like gene IDs, you have to click the features button here. You can't query both features and sequences in the same query. If you want to do that, you need to make a separate query or a new query. But I will walk you guys through that in a bit. So for the first query, we are just going to retrieve gene IDs and Go terms. So we click features here, then expand the gene button. We can see here it will automatically return, it will automatically give you gene ID and transcript ID in the table. We can expand the external button. We are interested in retrieving Go terms, so we can click the Go term definition, Go term name, Go term accession. And down here in the external references, we also have NCBI gene ID, so we can just click on that. If you're interested in external references, do be aware that you can only retrieve up to 3 for each query. So we have, as you can see here on the attributes table, this will return gene ID, transcript ID, all the Go terms, and the NCBI gene IDs associated with our genes. If we are happy with that, to retrieve our results, we go to the top left and hit the results button. And here, this is a preview of your results table. So here we have this column that is the gene ID. So these are the 6 different genes that have been returned by our filtering. Here we have transcript IDs associated with each gene ID. Here is the Go term definition, which is a very, very lengthy description of what each gene or each transcript does. Here is the Go term name. So it's just like a short, kind of like a short code, text code for describing what the gene does. And this is the actual code itself for this Go term. You can see here that NCBI gene IDs are empty, and that's because for wheat, a lot of wheat genes don't have an NCBI gene attached to it. But if you were looking at a different species like Arabidopsis, for example, you would more than likely retrieve a lot of NCBI gene IDs for your Arabidopsis gene. So by default, Biomart only shows you 10 rows, but you can choose to you can choose however many rows you want to be displayed to you here. If you want to save the results, firstly, I'll talk you guys through how to save it via email. So under this button, export all results to, if you click compressed web file, and then you choose the format of what you want, so you can either have it emailed to you via a web table like this, or a comma separated file, a tab separated file, or an Excel file. You then put in your email address here and press go, and that will email you your results. Alternatively, you can just save the file as is. So you just need to click File or Compress File, and then choose the format you want, and then press Go again. If you wanted to, for example, share this table with your colleagues or collaborators, you click this URL button in the top right, and it will take you to this link, and you can just share that link with whoever you want to, and then they will be able to look at the same query you've just generated. Okay, so I mentioned that you can only you can't fetch DNA sequences and things like gene IDs in the same query. So I will show you how to now fetch cDNA sequences. So we could just click new and restart the query, but that would be not the most efficient. So in fact, what we can do is just click on this attributes tab here again. And remember that we've selected all these things like a gene ID, transcript ID, Go terms under the features tab. If we now click on sequences, we can see that those attributes have been removed. And we are interested in finding the cDNA sequences. So under sequences, we can click cDNA. And it's important to know that you can only choose one type of sequence to retrieve per query. Here, we've ticked cDNA sequences, and you can see from this diagram, it shows you what the different sequences you'll be retrieving. So the cDNA sequences are essentially all the exons, all the coding regions of a gene. So we're only interested in retrieving cDNA sequences. So we've made sure that cDNA sequences has been included in the attribute section. Here, this is the header information. So this you can basically take whatever you want in here, and this will give you different bits of information for each sequence in your FASTA file. And this will make more sense when I show you the results. So we click results, and we can see here that it's generated a file for all the sequences for all the genes that we're interested in. So for all the 6 genes, each transcript has its own sequence associated with it. And if you remember, there was a header section just now I showed you, and that's what determines what gets displayed here. So this is a faster file. All the A's, T's, C's, and G's here are the actual sequence itself. The header information is denoted with a little arrow here. And then this is the gene name. The pipe is just a separator. And then here, this is the transcript version of the gene. So in wheat, there are usually three transcript versions per gene, but there may be more. And again, the principle is the same. If you want to save the data, so you can choose either save by file or have the data sent to you via email. But always remember that for sequences, you can only save as a faster option, which is this option you see here. So if that's it for the demonstration part of the presentation today. If there are any questions, do feel free to ask. If not, now you guys should have links to the exercises. So do feel free to complete the exercises, and we will be here if you have any questions regarding the exercises or if there are any questions regarding Biomart in general.
Thank you, Ian. Thanks, Ian, very much. So we have one question in the chat, and the person is asking how they can find genes associated with resistance to specific diseases. So I'm afraid that ensemble plants wouldn't be the go-to resource for this, but there are a lot of other resources that you can check instead. I have only one in mind, but Ian might know a few others as well. So we're going to post that into the chat so that you have it for reference. Are there any other questions for Biomart? Okay, so in that case, we're gonna begin with the exercises. So I'm gonna post the links into the chat again. But if you have the link already, you want to open the Biomart tab and continue with the exercises there. I'll give you about 20 minutes for this, and then we're going to come back together. And if there are any questions, or if you want us to demonstrate something again, or if you'd like us to explain anything again, please let us know. We have plenty of time today, so even if you have questions about the last few days, please go ahead and ask them now. So 20 minutes, and then we're going to move on. Hi, everyone. I hope you had a chance to try out the exercises for Biomart. So I can see there are a few questions in the living document that are outstanding. So Ian is going to answer them in the background while I keep talking. Do you have any other questions that you want answered live, or anything you want me to show on my screen? Okay, so in that case, I think we've pretty much covered everything we wanted to show you during the workshop. So I'm gonna share my screen because there are a few parting words I want to share before I let you go. Okay, so you should be able to see my slides. So before you finish up for today and get some rest and uh, process everything you've learned in the last couple days, I will ask you for one favor. So we always, after each workshop that we do, we ask participants to fill out a feedback survey. And this feedback survey really helps us to improve our courses for future participants, and also to find out if there are any bits in the workshop that you liked or less liked, or if there is any recommendations that you have about the website, for example. So if you have time now, please fill out the feedback survey. You can find a link on the training page. So I've switched to my browser, and in the training page, you can scroll down to the bottom of the list of the page and click on the survey link. So this is the survey. There are a few questions. It shouldn't take you more than 5 minutes, and I will give you time for that now. But at the very end of the survey, there is an option to receive a certificate of attendance. So if you would like to receive the certificate, just select yes and enter your email address into the next page. I'll share the survey URL with you now. And I will give you time to fill this out now. Hello, everyone. Welcome back. I hope you had a chance to fill out the feedback survey. If not, I will keep it open for another week so that you can fill it out in your own time. But for now, I'm going to move on, and I'm just going to summarize what we did in the last few days. So in the last couple of days, we explored the Ensemble Plants genome browser. And in the Ensemble Plants genome browser, you can find a lot of different genomic data, such as variation data, which we covered yesterday. We have different tools that you can use in your research. So if you have a list of variants yourself, for example, you can run it through the Ensemble vet and find out the consequences of the variants and how they affect the different genes and transcripts. You can also download whole genome sequences or whole genome annotation files from Ensemble through our FTP sites. So Ensemble is a very useful resource to find any kind of information about plant genes. Ensemble is what we also call an added value resource. So we call this an added value resource because it brings together information from a wide range of other databases into a single site. So remember, in your research, you wouldn't usually use a single source to get your information from. You would consult multiple different resources. So what Ensemble provides is links or entries to these other resources where you can start making your comparisons. Remember that Ensemble is just one of the many genome browsers out there. So another one you might be familiar with is NCBI's RefSeq. So RefSeq have their own annotation pipeline, and we have our annotation pipeline. So when you compare the two different databases, they may have some differences between the annotation, but remember that the sequencing is not done by us. So the sequencing data, the genome assemblies, are deposited by other researchers into public databases, and we take these public genomes and apply our own pipelines to them. So Ensemble data is organized in a very structured way, and there are different sites that you can visit. So as mentioned, we mainly looked at the Ensemble Plants browser, and this is part of the Ensemble Genomes website. So the Ensemble Genomes website is dedicated to non-vertebrate species. If you are ever going to work with vertebrate species, you can access the Ensemble main site. So this is just ensemble.org. And we also have what we call the Ensemble Rapid Release website. And this website is updated about every 2 weeks. And if you compare this browser with the other browsers, the data is a bit more limited. So the only data we have available in rapid release is gene annotation and some variation data and some comparative genomics analyses. And the only tool that you can use in rapid release is BLAST, whereas for Ensemble and Ensemble Genomes, you also have VEP, you can use Biomart and so on. If you want to use Ensemble data in your own pipelines, you can access Ensemble data via our Perl API and REST API, and you also have access to our MySQL servers. And again, if you want to download any whole genome data, please go to the FTP download site for that. If you try to download, for example, a list of all genes from wheat through Biomart, it's not going to work because Biomart will break. So if you want to get the whole genome sequence of wheat, for example, you can download that directly from the FTP site. Now, I mentioned in the first day that Ensemble data is completely open source. You can use it in your research as you wish. We do ask you to cite the most recent paper for Ensemble. So in this case, it would be the Ensemble Genomes 22 paper, and I've got the citation on my slide here. And if you enjoyed the course and you know someone or you have a colleague who might be interested in these kinds of courses, please do recommend us. So we've got the browser course, which we did in the last few days. Usually we would do these in a single day. It's quite intense. So it's actually quite nice to spread it out across multiple days like we did in this course here. We also have more advanced courses. So we have a REST API course where we teach you how to access the different data types through the REST API. And we also have a train the trainer course. So this is where we train you to become an Ensemble trainer. If you are interested in any of our courses or in hosting our courses, you can email us at helpdesk@ensemble.org. Or you can find more information in the training page. And there is a link on my slide here on the right. And if you want to reach out to us, there are different ways to do so. So we are on social media. We've got a Facebook account. We're on LinkedIn. We're on X as well. And we have a blog where we post any updates on the Ensemble data. We also post any job opportunities and so on there. And finally, some links to some help and documentation. So if you enjoyed this course and you want to learn more about genomics and bioinformatics in general, the EMBL EBI training portal is very, very useful for this. So there are a lot of open courses that you can do in your own time, and they're free. You can do them in the portal. If you're new to bioinformatics, for example, there is a very nice introductory course called Bioinformatics for the Terrified. So that's a really nice way to be introduced to the different resources that you can use. If you're interested in more Ensemble-specific tutorials, we have some more videos and walkthroughs on the Ensemble website. And we also have a YouTube channel where we post any short tutorial videos. And again, if you have any questions at all after the course, please don't be shy to email us. So we have a dedicated team in Ensemble who takes care of user queries. If you have any questions at all, please email us at helpdesk@ensemble.org. And we also have mailing lists that you can subscribe to if you want to keep up to date with Ensemble. And finally, I want to say thank you to Decode Live, especially for hosting this course. It's always great to work with you guys again. Of course, Ensemble is not just done by Ian and me. We are a very big team. There's about 100 people in our team, and we're a mix of researchers, developers, trainers, and so on. So it takes a big team effort to keep the website up and going and up to date as well. So thank you everyone very much for staying until the very end. I will share a link to the training page again with you. So the training page has all my slides and Ian's slides on there, and you'll have access to all of the links that I've just showed you on my screen. So if you have any more questions now, we will keep the chat or the Zoom call open for another 10 minutes. So if you have any questions, please feel free to ask them away. If not, I think you're free to go. And thank you all very much. Thank you, everyone. So yes, so I forgot to mention, there is still one outstanding question in the living document. I will answer that later today. So please keep an eye on it. If you have any other questions that you want to ask, I'll keep the living document open for another week as well. So you can ask away there, or you can email us at helpdesk@ensemble.org.
Louis, people are thanking you for your amazing sessions, and we also want to thank you for delivering 4 exceptional sessions during this plant genomics workshop hosted by Decode Life. Your expertise and dedication have significantly enriched our understanding of Ensemble Genome Browser. We are deeply grateful for your invaluable contribution.
Thank you very much. And yeah, if you want to, oh, oh, yeah, I just wanted to mention, if you're planning to do any future courses again, you know how to contact us. So we're always happy to contribute to your courses in future.
Sure, Louis. We will again invite your team for joint workshop or something which can be delivered on our platform. Thank you. Thank you for your efforts and contribution in this workshop. You are doing amazing service.
Thank you. All right, so I don't think there are any more questions that are being asked. So I'm happy to close the Zoom call. And thank you all again very much for staying until the very end. I hope that you found the course useful, and I'll see you soon, maybe.
Yeah, thanks, Louis, thanks, and Jamie team. And now I seek your permission for ending today's session with a hope to meet again sooner. Thank you.
Thank you.